All right. Well, thank you all for coming. So as Tarek mentioned, my name is Jesse. I'm currently a postdoc in the Bhatia Lab. So I previously completed my PhD in Sangeeta's lab. I graduated in 2020. And Sangeeta was benevolent enough to allow me to continue stopping by the lab when I had free time, now that I am a medical student. And so this work is some work that I started doing after graduating, sort of between graduation and starting medical school, and have continued since then. It's really a kind of a passion project of mine that Sangeeta has given me the opportunity to work on, for which I'm really grateful. So today we'll be talking about cholangiocarcinoma, which is a disease that most people have not heard of. And we'll be talking about developing non-invasive diagnostic tools to enable early detection of this disease. So what is cholangiocarcinoma? Well, cholangiocarcinoma is a malignancy of the bile ducts. So what are the bile ducts? Well, the bile ducts are a tubular kind of branching network of ducts that carry bile from the liver, where it's synthesized, to the small intestine, where the bile aids in the absorption of fats, among other functions. So cholangiocarcinoma can arise at any point along the biliary tree, from the intrahepatic or inside the liver portion to the hilar, which is kind of where the bile duct splits as soon as it enters the liver, and to the distal portion of the bile duct near the pancreas, and where the bile duct empties into the small intestine. Unfortunately, no matter where the disease is located, the prognosis is quite poor. And you can see that the overall 5-year survival rates for both intrahepatic and extrahepatic bile duct cancer is around 10%, which is on par with that of pancreatic cancer, which we all know of as one of the deadliest cancers out there. But one thing that you can notice when you look at this 5-year survival table is that whereas localized disease is by no means something that you would wish upon anybody, the outcomes are a lot better than for distant disease. But the majority of patients are actually diagnosed when the disease is already metastasized. So one thing that's important to note is, what can you actually do if the disease is detected at an early stage? So the Mayo Clinic has pioneered this protocol for actually doing liver transplantation for patients with cholangiocarcinoma. And what you can see here is that the outcomes for transplantation are vastly superior to resection. You can see that in patients who are successfully transplanted, so these are highly selected patients who have very early stage localized cancer that has not spread to any other part of the body. These patients are able to do quite well with over an 80% 5-year survival rate. And so our goal is to be able to detect more patients at the time when their disease is potentially curable by a curative method such as liver transplantation. But who would you even screen for a disease like this? Cholangiocarcinoma is quite rare. It wouldn't make sense to spend a lot of money and time and effort screening the entire population. And so it's important to think about who would be the population that would be screened for cholangiocarcinoma. And so there's a disease of the bile ducts known as primary sclerosing cholangitis, which confers a 400-fold increased risk for cholangiocarcinoma compared to the general population. And so I'm going to tell you a little bit about that disease. So primary sclerosing cholangitis is a disease of the bile ducts. The etiology is not well understood, but what is known is that it is the early stage of the disease is characterized by an inflammatory infiltrate around the bile duct, which is represented here in green, and then shown here histologically. And so there's a kind of a sparse inflammatory infiltrate, predominantly lymphocytic. And over time, this inflammation leads to injury, which leads to activation of stromal cells and the development of fibrosis or scar tissue. So this fibrosis or scar tissue is classically referred to as a periductal, so around the duct, or onion skin fibrosis. And you can understand by looking at the histology why this is called onion skin fibrosis. And the majority of patients will go on to develop cirrhosis and liver failure. But in about 20% of patients, this disease does end up developing into cholangiocarcinoma. And so patients with primary sclerosing cholangitis are screened on an annual basis with cross-sectional imaging for cholangiocarcinoma. But the early detection of cholangiocarcinoma, especially in patients with PSC, is extremely challenging. So this is what this is an MRI image of the abdomen. So just to orient you, so this is the liver, this is the spleen, and this here is a tumor that's encasing the bile duct. And so if a radiologist were to look at this scan, the radiologist would say this is a definitive cholangiocarcinoma. It's forming a mass that's encasing the bile duct. But most of the time in patients with PSC or other bile duct diseases that do predispose to bile duct cancer, the scan looks something more like this. And now just to orient you again, we have our liver, we have our spleen, and this is the bile duct. So take my word for it, this is not a perfectly normal bile duct. Actually, really, I'm taking the word of the paper that gave me this image, because I'm also not a radiologist. Actually, some of you might be a radiologist. I don't want to make any assumptions. But the bile duct here is not entirely normal. So it's slightly dilated. The wall is slightly thickened. But it's not definitive cholangiocarcinoma. It's not forming a mass. So what would you do if you were the doctor taking care of this patient? Would you immediately say, okay, you know what, we're gonna list you for liver transplantation. This might be cholangiocarcinoma. No, you wouldn't. And the reason for that is that in patients with benign diseases like PSC, the benign disease course can also present something like this. They can get inflammation of the bile duct, which causes it to dilate, causes the wall to thicken. So in patients who have a finding like this, it's a pretty bad scenario because they're in this sort of indeterminate space. They have to undergo an endoscopic procedure where a tube is inserted into the throat, snaked down to the small intestine. A little cannula is inserted into the bile duct, which is progressed up the bile duct. And usually there's like a brush that's inserted, and the brush is used to kind of scrape cells off of this suspicious lesion. Then those cells can be given to the pathologist who can do cytology, can do some other assays like fluorescence in situ hybridization to see if there's any chromosomal abnormalities. But even with all of that, which is invasive, it comes with a risk of pancreatitis from the instrumentation, it comes with a risk of infection. Even with that, the sensitivity and specificity of the cytology is actually quite poor, around 60 or 70% for detecting cholangiocarcinoma. So what we sought to develop in the Bhatia Lab is an imaging probe that could be coupled with existing diagnostics like MRI or CT to specifically light up malignant lesions without false positives from benign inflammation or fibrosis. And so for the rest of the talk, I'll be telling you about our efforts to develop a non-invasive diagnostic strategy to specifically light up cholangiocarcinoma lesions without false positives from inflammation or fibrosis. So in order to do this, we needed a target. And our lab has a long history of working with proteases, which are enzymes that catalyze the cleavage of peptide bonds. So they're proteins that cut other proteins. And why do we care about proteases? Well, proteases play a role in all of the hallmarks of cancer, from growth to angiogenesis, all the way to invasion and metastasis. And so the fact that proteases play a role in all these hallmarks of cancer and proteases are so dysregulated, that's led to the emergence of this new strategy, this new diagnostic strategy, which we call activity-based diagnostics or protease activity-based diagnostics, where proteases within the tumor, we're sort of like a pro-diagnostic agent, like a pro-biomarker can be administered into a patient who's suspected of having cancer. The proteases within that tumor are able to catalyze a reaction to change that pro-diagnostic into something that can be detectable, either an imaging-based readout like shown here, or a urinary readout as we've shown in previous publications, or a breath-based readout, a blood-based readout, et cetera, et cetera, et cetera. So there are some, so in addition to the fact that proteases are dysregulated in cancer, there are several other advantages that make proteases promising diagnostic targets. So for one, this is something that we all of us who have taken like a biochemistry course ever, you've heard the old trope that an enzyme is a molecule that can catalyze chemical reactions without using itself up in that chemical reaction, okay? And then, so what that means is that enzymes, proteases being one of them, have this self-amplifying property. A single enzyme can catalyze thousands of chemical reactions. So if you're thinking about a very small tumor, there might be very, very few proteases there. So instead of developing, for example, like an antibody that has, they can bind to a protease and release some kind of like a signal that can be read out to the rest of the world, by using a strategy that detects the activity of that protease instead of the presence of the protease itself, you can potentially increase, you can amplify the signal and the sensitivity hundreds or thousands fold. And then because we're engineers, we can develop tools that are modular. And by that, I mean, we can build these tools so that you can easily swap out different components of those tools in order to detect different types of proteases, have different types of readouts. Or potentially you can, deliver to different parts of the body, for example. And then lastly, protease activity is highly dynamic. One thing that we've shown in previous papers is that when you treat a mouse with a targeted therapy, within days, within like 2 or 3 days, you can already start to see changes in the protease activity, which is faster than you can get with morphological imaging, for example, like CT or MRI imaging. So moving into the technology itself. So the technology that we've been working with, the specific activity-based diagnostic strategy we've been working with, is what we call activatable zymography probes. And these probes are peptides. And they consist of, as I mentioned, they're modular, right? So they consist of 4 modular domains. So one is, this is a fluorophore, so it's a readout. The next is a polyarginine, so a positively charged domain that's in complex with a negatively charged peptide domain via a protease cleavable substrate linker. Okay, so this will make more sense in a second. But what you can do is you can take this probe, you can apply it, in this case, to tissue sections where proteases are active. And proteases within the tissue are able to cleave that protease cleavable substrate. Now, initially, this positively charged and negatively charged domain are they're complex together electrostatically, kind of like 2 magnets. But when they come apart, when that connection is severed, then that positively charged domain is able to bind to the many negatively charged molecules that are present in a tissue section or in a living cell. And so this strategy was actually originally developed by Roger Chen's group over at UCSD as a way to do non-invasive imaging, as a way to do in vivo imaging using this polyarginine domain as a cell-penetrating peptide. So polyarginine domains are well known to be able to bind to the cell membrane and then be internalized. So previously, as I mentioned, we've worked with ALK mutant lung cancer, and what we've shown is that when we apply these probes to tissue sections from this mouse model, we see this protease activity, which is Z1 in red, co-localizes with markers for endothelial cells, VE cadherin in yellow, and pericytes, desmin in green. And so in this particular instance, we found that this protease activity seemed to localize to vasculature, so endothelial cells and pericytes. And with single cell RNA-seq analysis, we learned that this probe was specifically detecting proteases that were responsible for neoangiogenesis. But you can, in addition to exploring the biology, you can also convert this into an in vivo imaging probe. And so when we administered this probe into mice with lung cancer, we found increased uptake in the tumor-bearing lungs compared to healthy lungs. So we sought to apply this strategy to cholangiocarcinoma. So the overall pipeline of the data that I'm going to show is we screened a library of 28 of these probes on tissue sections from cholangiocarcinoma. We performed some biological characterization, and we have made some progress towards developing an in vivo imaging probe for detection of cholangiocarcinoma. So first things first, we collaborated with a lab at Beth Israel, led by Yuri Popov, and he has a postdoc, Hinju Huang. And they had developed a mouse, the first ever mouse model of PSC, so this benign bile duct disease, associated cholangiocarcinoma. And so this model entails administering plasmids encoding for AKT and YAP1, two oncogenes, in mice with the MDR2 knockout. So without going into too much detail, these mice develop bile duct fibrosis, which resembles PSC. And you can see that these mice develop tumors that express cytokeratin 19, which is a marker for cholangiocarcinoma. So we screened 28 probes, as I mentioned. When we applied one such probe, AZP6, to mouse models from just this fibrotic mouse model, we saw no binding of that probe, so no yellow signal, and this is kind of the zoomed in version. This is a fibrotic bile duct. But in contrast, when we applied this probe to tissue sections from cholangiocarcinoma, we saw extremely strong binding that was specific to the tumors without binding to the region surrounding the tumor. And when we applied a broad spectrum cocktail of protease inhibitors, we saw complete abrogation of that signal, suggesting that there are proteases within the tumor that are cleaving that probe and allowing it to bind. So this is the quantification of that data. And I mentioned that we screened 28 probes. And for anyone here who's been to medical school, they might recognize that it would be hard as a medical student to do all of that by myself. And so I've been extremely lucky that I've had really great help in the form of John V, who is an undergraduate in the lab, who has done an amazing job and really exceeded expectations in moving this project forward. And what she's found is that there are actually 5 AZPs, so 5 probes in our library of 28 that bound strongly and specifically to cholangiocarcinoma tumors without false positives from fibrosis. So we next wanted to assess, are these findings specific to, or are these findings a model-specific artifact? So do they only apply to this one mouse model of cholangiocarcinoma, or are they generalizable? So we reached out to Daniela Sia, who is a professor at Mount Sinai in New York, who has developed multiple mouse models of cholangiocarcinoma. The caveat being that these mouse models are, these tumors are formed in healthy livers, not in fibrotic livers. But this was a way for us to get a sense of are our findings generalizable. And so specifically, we used some tissue sections from this mouse model, from this FBXW7 AKT mouse model of cholangiocarcinoma. And when John V applied one of those probes, AZP6 again, she found once again that the probe bound very strongly and specifically to the tumors in a manner that was protease dependent and without false positives from fibrosis. So indeed, this probe is generalizable across different genetic etiologies. So what is actually driving this biologically? So we first wanted to assess what are the cell types that are responsible for the cleavage of the probe. And so we performed a co-stain for AZP6 with cytokeratin 7, which is a marker for cholangiocytes or cholangiocarcinoma epithelial cells. And you can see this is the AZP6 signal in red, and this is the overlap. And when we performed cell-by-cell quantification, we found that AZP6 didn't actually seem to overlap very much with the epithelial cells. So there's a much stronger overlap there's a significantly increased binding of AZP6 to the cells that are low expressors of the epithelial marker. Whereas in contrast, when we looked at vimentin, which is a marker for mesenchymal or stromal cells, we see a significantly increased binding of the probe to the vimentin high or mesenchymal cell population. So then we asked, what are these mesenchymal cells? Are these cancer-associated fibroblasts? Are these epithelial cells that have undergone epithelial to mesenchymal transition? Are these endothelial cells like what we had found in our previous mouse model of lung cancer? So I'll just tell you, just so I don't have to go through all the data, not endothelial cells. We did a co-stain with endothelial cells. It's not endothelial cells. So that left a couple other hypotheses. So one thing we wanted to do to get at, could this be epithelial to mesenchymal transition that we're seeing? We know that that's a hallmark of cholangiocarcinoma in patients. Could that be what's going on here? And so to start getting at that, anyone here who has, who's an EMT expert, please forgive me for my extremely rudimentary approach here. We just did a co-stain for an epithelial cell marker, cytokeratin 19, a mesenchymal cell marker, vimentin, and we sought to see, are there cells that have an intermediate cell state? So this is the vimentin stain. This is the cytokeratin 19 stain. This is the overlap. And if you zoom in, you can see that there are several cells here that do stain positively for both the red signal and the green signal, leading to a yellow signal, suggesting that there is some kind of an intermediate cell state. It does not prove EMT, so we haven't done any formal lineage tracing. Those are kind of some ongoing studies that we're interested in the future. But it is a sign that these might actually represent that there might be some amount of EMT that's going on here. So that's the cell type. What about the actual protease that's responsible for the cleavage? So one thing that we tried is to use protease class-specific inhibitors, okay? So there are 5 different classes of proteases: metalloprotease, serine, cysteine, aspartic, and threonine. The most abundant classes of extracellular proteases, so proteases that are secreted, are serine and metallo. And so we attempted to apply inhibitors specific to either broad spectrum proteases, serine proteases, or metalloproteases. When we apply broad spectrum proteases, we see complete abrogation of the signal, as we had previously shown. But when we applied only inhibitors against serine proteases, once again, we see complete abrogation. But when we apply inhibitors to metalloproteases, we see minimal to no abrogation of the protease activity. And we can quantify this here. And so this suggests to us that the protease that's responsible for the signal is serine protease specific. And one thing that Janvi has done recently is to take all 5 of the probes that we've tested, that we found to be hits, that are specific to cholangiocarcinoma. And she's done this protease inhibitor study. And she's found that for all 5 of them, so this is the 4 plus the one that I showed in the previous slide, the signal is either significantly or substantially, because there were not enough, the N was not high enough to quantify here, decreased signal when we apply serine protease inhibitors. And we now have some follow-up studies where we're looking to figure out what the specific protease is, not just what the specific class is. So hopefully the next time I give this Marble talk, hopefully this paper will be published. But if it's not, then we'll give you some more updates on what the protease is. And so lastly, we wanted to adapt this probe for PET imaging, to adapt this probe for in vivo imaging. We're not at PET yet, but I can show you some pilot proof of concept data that this might work. So we modified that probe, AZP6, by adding a quencher to make the signal more specific, and then by pegylating it to increase the solubility and half-life. And we administered into either the tumor-bearing mice or fibrotic mice. And then we took out the livers 2 hours later and we imaged. And what we were pleased to see was that in the mice with cholangiocarcinoma, we did see a small but significant increase in the signal compared to the fibrotic mice. Now the tumor burden was extremely small here, and we hadn't optimized the kinetics of the imaging timing, but it was a positive signal to us that this might work. And so that's where I'm going to leave the data today. But to conclude, we've screened 28 AZPIT and 28 AZPs, identified 5 that are cholangiocarcinoma specific in two genetically distinct mouse models. These AZPs are cleaved by serine proteases, and they seem to co-localize with a mesenchymal cell population that's possibly derived from EMT, possibly. And then we also find that there is increased uptake in tumor-bearing livers after in vivo administration. So the next steps we're really excited about are, 1, to adapt this probe for PET-based imaging. And the reason for that is that fluorescence is well and good if you're working with a mouse and you can, perform explants and things like that. But what you really want to do is have a probe that could be used in the existing clinical workflow. And if you have a suspicious lesion, you could just be like, okay, we'll do a PET scan now. And that could tell you with confidence whether this is benign or requires further workup. And so that was what I alluded to in the kind of the pipeline of this talk. And so what we've done so far now is we've synthesized a probe that has a chelator that we could combine a radiolabel with, and we're working with Howard Mack at the preclinical testing core here at the Koch. And then lastly, we're excited about converting these, or we're excited about also thinking about translationally testing some of these probe hits in human cholangiocarcinoma, which we're sourcing through a collaboration with the University of Birmingham in the UK. And so with that, I will conclude. I want to give a huge thanks to Sangeeta because she has put up with me for the last 8 years now, which is crazy. I think that Sangeeta has created an extremely innovative and productive, but also educational and supportive environment in her lab, which has been so crucial to my development as a scientist and as a person. And I'm so grateful for the opportunity to be working especially on this project in her lab. I've already given a shout out to John V, but I want to reiterate just how talented of an undergraduate she is, and how grateful I am that she's been able to move this work forward with my spotty attendance in lab. And lastly, I want to give a big shout out to Vardman, who has provided a lot of mentorship to me and to John V, and has also helped keep this work going. There are many other collaborators that I want to acknowledge, but I will spare them the embarrassment. I will just give a shout out to the Popov Lab, the SIA Lab, University of Birmingham, and the core facilities here at the Koch. I want to also give a big thank you to the Marble Center for supporting this work, as well as to the Resnick Family Center at Brigham and Women's for their contribution. And thank you all for your attention.